allows the muscle to shorten. So what's going to happen is these two proteins are going to interact with each other. They're going to slide past each other, and they're going to essentially shorten the entire sarcomere. Now, the shortening of the sarcomere is what brings about muscle contraction. Okay? So that's really what we're going to look at today. Now let's look at the structure of those two proteins. So we said the thin filament formed by actin. Okay, the thin filament is actin. And that is essentially formed by several G-actin monomers. Okay, so several G-actin monomers. This is a G-actin. And G just means that it's a glomerular protein, right? And it has a binding site for myosin, which is the thick filament, to attach here. Now, when these monomers line up together, they form F-actin. And F-actin means that it's now a fibrous protein, or it's forming a filament here. Okay, now two F-actin molecules are going to intertwine around each other. Okay, they're going to wrap around each other to form this double helical actin strand. Okay, let me repeat that. We had monomers of G-actin lining up together like pearls on a string to form F-actin. Two F-actin molecules wrapped around each other to form the double helical actin strand. Now, actin is also associated with two regulatory proteins. The first one is tropomyosin, and that is going to cover or hide these binding sites. It's going to wrap around that actin strand, and it's essentially going to cover up or hide, right, the myosin binding sites in such a way that when the muscle is at rest, we do not want actin to be exposed for myosin to bind. Okay, so tropomyosin covers up or hides or prevents the binding of myosin. And the second regulatory protein is actually a complex of three proteins. Right? There are three areas for this complex to bind. The first one is going to bind is the tropomyosin. It's going to bind tropomyosin. The second area it's going to bind is the actin monomer, or the actin fiber. And the third site that's on the troponin for binding is for a calcium. Right? A calcium molecule is going to bind onto this troponin complex, and that is going to initiate muscle contraction because that will allow tropomyosin to uncover those binding sites. Okay, is that clear? So I'll repeat that one more time. Calcium will bind onto this troponin complex. That will cause a shift in that protein, which will then unbind or unblock the myosin binding sites, and myosin will be free to engage with actin. Question? Okay, so tropomyosin is blocking or hiding the binding sites, and this is in the absence of calcium. Okay, this is why calcium drives this process so much. When calcium is not present, tropomyosin is hiding those binding sites. As a result, these proteins cannot interact with one another, right? Because this binding site is covered. Calcium will come in, right, when it's released, and we'll look at that mechanism later on, bind onto the troponin complex. Now, because one of these sites, or one of the areas of this complex, are also binding tropomyosin, it's going to sort of shift the protein off of those binding sites, exposing the binding sites, and now myosin can freely bind onto actin. Okay? So in that way, these proteins are regulating this process because we do not want actin to continually binding to myosin. Again, we want to regulate muscle contraction because there are times when the muscle needs to relax. Okay, so as a result, these two proteins are regulating the binding of actin and myosin. All right, now let's look at the structure of myosin. Myosin is forming the thick filament, as we saw, which is the center of the sarcomere, essentially. Okay, and it's going to be, its tail is going to be facing the M line, which is the middle of the sarcomere, and its head is going to be reaching out towards the Z line or towards the I band. Okay. Now you can think of myosin as two golf clubs essentially wrapped around each other. Okay. And the head of the myosin has two binding sites. One is going to bind actin. Okay, so we saw the myosin binding site on the actin molecule. And here is where it will bind on the myosin molecule. So those two proteins will come in contact at these binding sites. And the second binding site here is for ATP to bind. Okay, ATP is necessary for this process, right? And it's going to bind onto the myosin head, which is going to change the configuration of the thick filament. And we'll see how that happens. There's another protein that we're going to look at, and that is called titin. Titin. And titin is a very stretchy, very elastic protein. And it's essentially going to support the muscle when this contraction or shortening is happening. So titin is going to bind to the tips of the thick filament and stretch all the way to the Z line. Okay, so it's essentially going to stand between the thick filament and the Z line, and it's going to shorten as the sarcomere shortens, because it's a coily or stretchy protein. And it's also going to stretch as the sarcomere relaxes. So having that stretchy protein, right, interacting between the thick filament and the Z line is what allows the sarcomere to shorten and lengthen, which produces muscle contraction. Okay. All right, so let's look at a question. So what is that long fibrous protein that extends over the actin monomers and blocks the myosin binding site when muscle is in its relaxed state? What was that protein called? Tropomyosin. Perfect. Tropomyosin. Okay. Let's look at one more. So this is asking for the name of the dark striation observed under the microscope that is due to the overlap of thin and thick filaments. The A band, correct. The A band was that area that had the overlapping of the thin and thick filaments. All right, so we're going to move on. Any questions before we get moving on? We're going to move on to talk about the sliding filament model. Okay. And that's essentially how these two proteins interact with one another to bring about muscle contraction. We're also going to talk about the cross-bridge cycle. Okay, the cross-bridge cycle. Now those little heads that I drew sticking up, just go back here quickly. Right? These little heads that I drew sticking up here, these are called cross bridges. These are called cross bridges. And essentially, that's where the myosin head is sticking out to reach up and grab on to the actin. Okay, so we're going to talk about how those two structures come in contact with each other. Now, when the thick and thin filaments overlap, right, and the sarcomere shortens, the early researchers who started studying this process used to think that the thin filament contracted, right, or the thick filament contracted. And now we know that that is not true, right? They do not contract or shorten on their own. What happens is they slide past each other. So the length of the actin and myosin is not really changing, but because they're sliding past each other, and we're going to see how that happens, the entire sarcomere is shortening. Okay. All right, so the sliding filament model is what happens when the sarcomere shortens. And we'll see several changes in the bands that we described earlier. So first of all, the I band. And just to remind ourselves, we said the I band is the area that only contains thin filaments, right, or actin. And as myosin slides this way, right, the I band is going to shorten. So with the shortening of the sarcomere, the I band shortens. The H zone will also shorten. So the H zone is the area, this area here, which only has myosin, right? Only has thick filaments. And again, as these two proteins slide past each other, the I band or the H zone will shorten, right? It's in the middle, and myosin is going to slide under actin. It's going to shorten that central area where there's no overlap. Okay. Now, what is not going to shorten is the A band. Okay, the A band is going to remain the same. Why? When we look at the sliding, so myosin is sliding that way under actin. Actin is being dragged, okay? And as a result, the movement in both directions cancels out. So the I band, okay, which is the area of overlap, the A band, keep messing up my words. The A band, the area of overlap, is going to remain the same, okay? So the I band shortens, that's your thin filaments. The H zone shortens, that's your thick filaments. And the A band, which is the area of overlap, will remain the same, okay? Now, when this process of shortening happens, right, when this shortening of the sarcomere happens, there's gonna be a continuous contact and breaking. That's called the cross-bridge cycling. And cross-bridge cycling means that actin and myosin are going to come in contact. Myosin is going to drag actin along. They're going to break contact, and it's going to go back and start that process again, okay? Which is what shortens the sarcomere,